Great, thanks, Mitch. I'll just need to work out how to scroll on this slide. There we are. Following on from Mitch's discussion about the importance of reproducibility and also for synthetic gels, I'd like to tell you a little bit about the work we've been doing in our laboratories here in Glasgow in conjunction with Biogelx. Particularly, I want to talk about two things. The use of peptide hydrogels to direct stem cell differentiation. This was not developed in Glasgow, but we developed a little turn on this that I think is important for synthetic gels, and that is trying to direct stem cell differentiation and also look a little bit about possible discovery of drug types. And then the use of peptide hydrogels to improve the cartilage phenotype in 3D culture, which again is a big regenerative medicine challenge. So for the first story, I'm going to focus on the use of pericytes. So let me first explain what these are. So these are perivascular stem cells, which are considered multipotent precursors of mesenchymal stem cells, which of course most people have heard of now. Pericytes are found on the vasculature throughout the body. They're multipotent as a mesenchymal stem cell, so can turn into osteoblasts, chondrocytes, adipocytes, fibroblasts, etc. And they were first published as multipotent precursors of MSCs in this paper in Cell Stem Cell from the Pio group over in Edinburgh in 2008. And we wanted to observe differentiation on different gel stiffnesses. It started as quite a casual study. We wanted to look at the very well-known Adam Engler, Dennis Disher study published in Cell in 2006, where they put mesenchymal stem cells onto gels of 3 stiffnesses. They had a very soft gel around about 1 kilopascal, a medium stiffness gel of around about 15 kilopascals, 10 to 15 kilopascals, then a stiff gel, a rigid gel, up to around about 40 kilopascals. And on the soft gel, around 1 kilopascal, they saw expression of neural markers from mesenchymal stem cells. On the medium gel, the 10 to 15 kilopascal gel, they saw myogenic expression, muscle cell expression for the mesenchymal stem cells. And on the very rigid gel, the 40 kilopascal gel, they saw expression of the osteoblast phenotype. Now, just a little aside, the observation of neural expression markers is kind of a moot point. And personally, I believe this is the expression of stem cell self-renewal markers. So the bone marrow, as for neural tissues, is very soft. It's just less than about a kilopascal in stiffness. And mesenchymal stem cells or perivascular cells within the bone marrow will express neural markers like beta-3 tubulin and nestin. And these are usually considered to be when they're homing hematopoietic stem cells. So they share the bone marrow niche with the hematopoietic stem cells, and the mesenchymal stem cells and pericytes are thought to have a supportive role. And actually, Bruno Pio and his group have shown that if you culture hematopoietic stem cells over pericytes rather than mesenchymal stem cells, you get better self-renewal of the pericytes. But this is a whole different story for a whole different day. But the important thing is we could take 3 stiffnesses of gels, the soft, the stiff, and the rigid, and we could differentiate the cells. And for the soft and the rigid, we saw the same as Engler, with the pericytes turning into cells expressing neural markers on the soft gel. And here you can see expression of beta-3 tubulin in part C of the image on the right. And on the far right in part E, on the rigid gel, you can see expression of bone markers, in this case RUNX2, here, indicating differentiation towards osteoblasts. We differed in the middle. Whereby when we used our stiff gel at 13 kilopascals, we didn't see myogenesis. We saw expression of chondrocyte markers, and we've given an example here in part D of SOX9 having increased expression. So for the most part, we agreed, but apart from this one in the middle. Okay. So what we wanted to do, the big advantage we had over other groups at this time was that we're using a gel source that were very similar. So we had 3 highly controlled rigidities, and that's really important. Using many material systems, controlling rigidity or stiffness with good level of control is quite tricky. With the Biogelx gels, it's pretty straightforward. But because the gels are synthetic, and the only thing that's changing is the fiber density of the peptide fibers, actually they're chemically very similar. We don't have to coat the gels to get the cells to stick. They'll stick onto the native gel. So there's no intermediate layer to add complication, and we don't have to use cross-linking agents such as glutaraldehyde to get the most stiff gels. And this was a big advantage because at the same time my group's getting into looking at metabolites and metabolomics using mass spectrometry. And we wanted to find metabolomically quiet material systems to tell the cells what to do, but not to affect the metabolites themselves. So we just wanted stiffness to tell the cells what to do, not the chemistry or the cross-linking agents that we're putting into the gels. So as the cells differentiated, and here I'm just going to present on chondrogenesis and osteogenesis, because the neural story, whilst it worked, is more complicated, and so it's better left when there's longer time. What we did, we took 2 time points, an early time point and a later time point in the differentiation process for chondrogenesis and osteogenesis, so on the stiff gel and on the rigid gel, and we followed the metabolome with time. We looked particularly at the lipid metabolome, because this is where we got the most hits. And it's an energy-dependent, these are energy-dependent metabolites, so and differentiation's an energetic process, and so energy seems a good place to look. And what we look for was unique depletions with time. So metabolites the cells seem to be using to differentiate. And so at the top of the figure in part A, we can see that this metabolite, GP18-0 or lysophosphatidic acid, is being used by the cells when they undergo chondrogenic differentiation, so on the middle stiffness hydrogel. Whereas it's not found whereas cholesterol sulfate, which I'll tell you in a second, is not found in this gel. So this GP18-0 is a unique depletion. When we look at the rigid gel, so the Pericytes undergoing osteogenesis, we see the opposite. There's no change in GP18-0, so this isn't part of the osteogenic process. But on for cholesterol sulfate, we see this depletion between week 1 and 4, which suggests this is being used during the osteogenic differentiation process. And if we use pathway building software, we can see that GP18-0 is a glycerolipid, whereas cholesterol sulfate is involved in steroid biosynthesis. So all we've done is we've used the gels to let the cells differentiate, and we've just looked at what the cells are using to differentiate in terms of metabolites. And metabolites, as I'm sure you all know, are the small building blocks of biochemistry, so little bits of biology that cells use to drive their biochemistry as they differentiate or as they grow. So the next thing we did was culture the cells just on glass cover slips. What we wanted to see was, could we take these metabolites the gels had showed us, and could these metabolites then themselves drive cell differentiation, just individual metabolites? And here we use both pericytes, as you can see in panels A and E, and mesenchymal stem cells, as you can see in C and G. So on the left hand of all these images, you can see the control cells. So these are just pericytes or mesenchymal stem cells cultured on a cover slip. In green are phenotypical proteins. So on the left, we can see osteopontin and osteocalcin. These are proteins expressed during bone formation. On the right, we can see aggrecan and collagen 2. These are proteins expressed during chondrogenesis or cartilage formation. So if we take the figures on the left to start with, we can see the control cells where there's no green being expressed, so no bone marker proteins being expressed. Then in the middle, we use osteoinductive medium, OIM, which is dexamethasone, glycerophosphate, and ascorbic acid to drive bone formation. You can see good expression of osteopontin and osteocalcin in both the pericytes and the mesenchymal stem cells. But then in the last column, we've just used cholesterol sulfate by itself. So this metabolite that we identified just by following the fate of the cells on the hydrogels. And we can see that we're getting similarly efficient expression of the bone phenotype, the osteogenic phenotype from stem cells, as if we used the more complex and standard osteoinductive media. Remember that dexamethasone is a powerful corticosteroid. Over on the right, in the right-hand panel of images, this is the chondrogenic panel. We can see again on control, on the left image of this right-hand panel, we see no expression of aggrecan or collagen 2, these chondrocyte markers, whereas in the middle, where we use chondroinductive media, and this contains transforming growth factor beta, TGF-beta, insulin, and dexamethasone, so corticosteroids and growth factors to drive the chondrogenic phenotype. Whereas over on the right, where we've just used this single metabolite, GP18-0, we can see similar levels of chondrogenesis in the stem cells, both the pericytes and the mesenchymal stem cells, as when we use the very complex, expensive chondroinductive media. And we find this kind of interesting because things like dexamethasone and growth factors, they can have off-target effects when they're used in the body in vivo. And so single metabolites that are found in vivo anyway might seem a simpler, more cost-effective, and potentially safer way of inducing different phenotypes in vivo. So the take-home message from this first study is that these gels we've used are pretty much identical. We don't have to differentially cross-link. We don't have to use chemicals to induce differentiation or coat with proteins. This has to be done with, for example, acrylamide systems. This means that they are metabolomically quiet, vital to allowing us to look for the metabolite differences caused by the gels alone. This study gives us a demonstration that gels can be used, that biomaterial systems can be used to persuade stem cells to do things, so we can watch them and identify bioactive metabolites that could potentially go on to be developed as drugs to control stem cells. And this work was published in Alakpa et al. In Chem in 2016, tunable supramolecular hydrogels for selection of lineage guiding metabolites in stem cell cultures. So moving on to the second story, chondrocyte tissue engineering. Now, with an aging population in developed countries, we're particularly worried about musculoskeletal disease, arthritis in particular, because it costs, it has a large socioeconomic burden, people are in pain, we're all living longer, and as we have this increased lifespan, we also want to enjoy increased health span to go along with this. So we have to think about regenerative medicine approaches to helping solve problems such as arthritis. And because the stiff hydrogel, the medium stiffness, 13 to 15 kilopascal hydrogel, could induce chondrogenesis without using differentiation factors, we decided to have a look at this. This time, I should say in the previous study, we were putting cells onto the hydrogels rather than in the hydrogels. We're just using the substrate, but here we're putting cells within the hydrogels as a three-dimensional tissue engineering construct. I should mention we didn't use a solid 24-well plate-fitting hydrogel as shown in part B. We used droplets of hydrogels, so little spheres of hydrogels with the pericytes inside. So we're still sticking with the pericytes here. We should note that there's some parallels between the stiffness of the hydrogel and the stiffness of chondrons that chondrocytes grow on in the Cartage environment anyway. So when we encapsulated the pericytes, we saw, as we can see here in part A, very good cell viability. All the cells were alive over all the time course we did, up to 168 hours. And further, by qPCR, as we can see in part B, we saw enhanced expression of chondrocyte markers SOX9 and collagen 2 after 1 week of culture. And if we look at histology after 28 days, we can see in part C and D high levels of expression of aggrecan, as shown in C, and collagen 2, as shown in part D, both stained green with the nuclei stained blue within these hydrogels. So we can see we're getting both viability, survival of the pericytes, and differentiation of the pericytes in 3D, as we'd seen on 2D with the stiff hydrogel. So we wanted to move on to looking at different markers, and so we moved to using qPCR at various time points from day 1 up to day 35. Here we used planar control, which is just a cover slip. We used alginate as a typical material that chondrocytes are encapsulated in, a gold standard if you like. We've used the stiff hydrogel, which is denoted here in the grey bars as FMOC F2S. And we've also used the hydrogels, but with chondroinductive media. So that's denoted with a positive sign. So just the cells in the hydrogel alone with the negative sign, cells in the hydrogel with chondrogenic media with a positive sign. And what we see is in the hydrogels, compared to alginate, compared to cover slips, we get much better expression of SOX9, of collagen 2, of aggrecan, and of type 10 collagen, as would be expected for cells undergoing chondrogenesis. And really importantly, much higher expression of cells in alginate in 3D. As you can see by comparing the grey and the yellow, the hydrogel bars to the orange alginate bar. But whilst it was noted that adding in the chondroinductive media increased chondrogenic differentiation further, it's important to note that the hydrogel itself, the stiff hydrogel itself, drove chondrogenesis just fine. Actually, it drove it rather better than the using chondroinductive media with the hydrogel. If we look at the ratio of proteins, So we looked at GAG to collagen 2 and also collagen 2 to collagen 10. Now, the important one really is to look at the collagen 2 to collagen 10, because if we consider the structural function, the structure of cartilage, at the top you've got the superficial zone. This is the articulating hyaline surface of the cartilage that glides and allows the joints to move, and this is what gets worn out during arthritis. At the deep zone of cartilage, close towards the bone, you've got hypertrophic chondrocytes and mineralized cartilage as it interfaces with the bone. Now, at the top, at the articulating surfaces, the cells should be high in collagen 2. At the bottom, at the collagen 10 surface, at the bottom surface, the bone interfaces should be high in collagen 10. So if you look at the graph in part B here, you can see that the cells grown within the stiff hydrogel alone, without the chondroinductive media, have a much higher collagen 2 to collagen 10 ratio, which means we're getting this more hyaline articulating cartilage phenotype within the gel compared to using TGF-beta in the inductive media. And it's very well known that TGF-beta causes overexpression of collagen 10 in cultured cells. So this is quite an important observation. So if we look at the metabolome, we can see very clearly on this figure below, if we see the planar cells grown on planar samples compared to cells grown within the hydrogels, very large difference in the kind of heat map from looking at the metabolome, the cells from planar versus undergoing chondrogenesis. The cells within the gels, but with and without the chondroinductive media, look pretty similar, except for discrete pockets of differences, as you can see outlined by the dotted lines. Now, it's within these pockets that we're researching now, because this must be the areas where we have the difference between collagen 2 expression and collagen 10 overexpression. So we have to research these more deeply as we go forwards. But for the last couple of slides, just to summarize, for techniques such as matrix-assisted autologous chondrocyte implantation, or MACEs, I'll quickly show in the next slide, use of TGF-beta in differentiation causes chondrogenic cells with high collagen 10 expression, typical of deep zone chondrocytes. So that's not what we really want when we are repairing cartilage. The gel alone is allowing us to generate chondrocytes from stem cell populations with higher expression of type 2 cartilage, typical of hyaline articulating cartilage, and this work was published in the LACPA in Scientific Reports in 2017, improving cartilage phenotype from differentiated pericytes in tunable peptide hydrogels. So just for the last slide, it's just a picture of the structure of cartilage. On the left, we can see the part 1, the superficial zone. This is the articulating zone. Then at the bottom, you've got the larger hypertrophic chondrocytes, high in collagen type 10 in the calcified zone, whereas in the superficial zone, you have a higher amount of collagen type 2. In the middle, from the polymers reference, this is a figure showing MACE, matrix-assisted autologous chondrocyte implantation. So on the knee, you can see a cartilage lesion. They take a biopsy from the non-articulating part of the cartilage, expand these up in culture as chondrocytes, then they can inject them back into the wound site. That's an autologous chondrocyte implantation. Or they can use a polymer to fill the wound site first and then inject the cells in. And this is a matrix-assisted autologous chondrocyte implantation. They put a little patch over this. This is a shaved patch of the periosteum. There's an oval patch depicted on the bone just below. They shave that off, it's like a skin, and they suture that over the defect. And on the far right, you can see an image of Cartage patch being put into place within a knee. And this is an alginate aggro scaffold. And we can see from our data that alginate doesn't push the cells to become chondrocytes, so it doesn't support them to chondrocytes. So if we think about towards the future for regenerative medicine using defined hydrogels with a stiffness that tells the cells what to do, we can probably get a much better repair going forwards. So that's all I want to tell you today. Thank you very much.